All right, everybody, let's get today's program started. Good afternoon, folks. Welcome again to the Lunchtime Discovery Series. It's good to be with you for today's program. We had a little bit of a break last week, so I haven't seen y'all in a few days, or you haven't seen me, I guess it is. But I hope everybody had a great long weekend, if you got one, that is. If you celebrated the Thanksgiving holiday, hope you had a fantastic time. We are glad, though, to be back here on your screens for the Lunchtime Discovery Series, bringing you interesting people doing interesting work out there in the worlds of science, nature, uh, education, art, and more. Today, we've got a great program for you. I think you're going to like it. Actually, a return guest to the Lunchtime Discovery Series today. Now, the program is brought to you, it's organized by the folks over at the Office of Environmental Education within the Department of Environmental Quality, and then broadcast by us here at the North Carolina Museum of Natural Sciences. I am your host, Chris Smith, as always, or almost as always, and I'll be helping facilitate the Q&A after today's guest speaker delivers their presentation. So make sure you all stick around. Make sure that you have those questions queued up in the chat for me. If you got a good question, pops into your brain, drop it into the chat. That way we can have a fantastic conversation about the topic of the day. But I shouldn't hold up why you are all here. Everybody, I would love for you to meet Dr. Stacy Zhang. Dr. Zhang is an assistant professor at the Department of Marine, Earth, and Atmospheric Sciences over at North Carolina State University and is a repeat guest here at the Lunchtime Discovery Series. Did such a fantastic job, we had to have him back. Everybody, say hi to Stacy. Welcome.
Thanks. I appreciate that introduction, and I'm glad to be back. As a North Carolina native, I grew up going to the museums, and so it's fun to be a part of the whole process and sharing my science. So, all right, shall we get started?
I think we should. Let's do some learning.
Oh, okay. All right. Okay. So I'm going to chat with y'all today on some really cool science that my lab has done in the past, but also things that other researchers have really advanced the science in terms of beneficial partnerships in ecological communities. And so if I just start with this photo that you see right here, it doesn't look like much probably when you first glance at it. It probably looks like a lot of brown and a little bit of green, but what you can hone in on if you look closely is there's a bunch of mussels and oysters growing along with this grass. And this is all a beneficial partnership that is essential for the formation of our local salt marshes. And hopefully by the end of this talk, you'll be part of the same crowd that I am, where I love the concept of beneficial partnerships. So I'll start sort of by giving a quick summary of the last time I was here. I talked about hurricanes and their impacts on coastal fishes in North Carolina. And one thing I talked about was about community resilience. And sort of as my segue into this talk, one of the things that I really learned on a personal level, having experienced many hurricanes growing up in North Carolina and then moving down to the coast, living through Hurricane Florence, was not just the massive impact of these storms, but also the amazing way that the community comes together and helps each other in response to these storms. And being a part of some of those response efforts was absolutely life-changing for me, and seeing the way that, people that don't even live in the area are sending resources, sending trucks full of water and food and tarps for people's houses. People are on each other's roofs, fixing the shingles. This amazing way that a community comes together in response to disturbances. And I kind of see this as sort of a underlying theme in a lot of the work that my research has taken in the last couple of years. And this is this concept of teamwork. And so I like to say that the cheesy tagline of some of my research is that teamwork makes the dream work, and that there are all these ways in which organisms are working together in order to form the ecosystems that we can see with our naked eyes. And so I love this photo of Space Jam. For many of you, you probably are more familiar with the more recent one with LeBron James, but this will always be my go-to reference, no matter how many years pass. But most of us, when we think of ecological partnerships, we may think about symbioses that we were taught when we were younger in grade school. And these are close, prolonged associations between two or more different biological species. On a very basic level, what we were probably taught were things like mutualisms, where both species benefit. And so, one of the key examples of this is pollinators and bees pollinating flowers. But also parasitism, where one species derives nutrients from another, so that would be the host at the host's expense. And so there are a lot of different examples of this. I think I remember being taught the examples of tapeworms, but there's also fungi that will infect ants, and a lot of cool examples across the ecological world, as well as commensalism, which is when one species benefits and the other experiences no net effect. So in this example, this is the case of two actually parasitoid wasps that live together, and in the absence of one wasp species, the other doesn't experience a negative effect, but one of the species does actually gain a lot of positive benefits from the other. And so it actually is a little bit more complicated than this, but I won't bore you too much with going into all of the specifics. But what is really interesting about symbioses is how much it has come to the forefront in terms of research in the last couple of years. So much so that it's actually becoming a part of a lot of the media that we consume. And so I'm going to share with you a couple of really cool clips from television shows that have incorporated symbioses in the last couple of years. And so this first one is from a show called The Last of Us.
And that's your biggest worry?
Yes. Also a fungus. Viruses can make us ill, but fungi can alter our very minds. There's a fungus that infects insects, gets inside an ant, for example, travels through its circulatory system to the ant's brain and then floods it with hallucinogens, thus bending the ant's mind to its will. Fungus starts to direct the ant's behavior, telling it where to go, what to do, like a puppeteer with a marionette. And it gets worse. The fungus needs food to live, so it begins to devour its host from within, replacing the ant's flesh with its own. But it doesn't let its victim die, no. It keeps its puppet alive by preventing decomposition. How? Where do we get penicillin from?
Fungus. Oh. Dr. Schoenheis, you're in distress.
Fungal infection.
So I actually haven't watched The Last of Us, but this is a show that my students have told me about that has talked a lot about fungal networks. And the example that the person in this video is talking about is actually the photo from the last slide of that ant and the little fungus that's growing out of this ant. And so this is a reference to an actual parasitism relationship, but of course it's dramatized for television. But one of the ways that this fungus in this show communicates is that they are able to send messages amongst the network of different fungi. But that also,
and that's your biggest worry.
Yes.
By another ecological relationship, and that is mutualisms. And so this is a clip from one of my favorite shows, Ted Lasso, that talks about fungal networks in trees. Let me scoot forward.
Hi, I've got this. We used to believe that trees competed with each other for light. Suzanne Simard's fieldwork challenged that perception, and we now realize that the forest is a socialist community. Trees work in harmony to share the sunlight. Can't you just give me a straight?
So without going into too much detail about Suzanne Simard's work, but it is a really cool field of research that's been exploring how these mutualisms that we maybe learned about when we were kids between mycorrhizae, which are the fungi, and roots, are critical for ensuring that our tree systems and our forests can actually function. I think many of us were probably taught about mycorrhizae and George Washington Carver and the impacts that these fungal symbioses have for farming, but it actually goes way beyond just that one case. And so what I will say is that a lot of the work these days is really pushing more towards understanding these positive interactions rather than competitive or predatory interactions. We're seeing this emphasis on beneficial interactions. And so these are examples like direct facilitations. And so just to give you a couple of simplified diagrams, this is where you can have one species that directly benefits another, or you can have one species that directly benefits multiple different species. And this is sort of like a whole ecosystem facilitation. There are also examples of mutualism. So similar to the example I showed with the bee and flowers, where species A will benefit B, and B also positively impacts A. But there's also a grouping of indirect positive interactions. And so these can either be double negative or double positive. And so this is an example where, for example, species A will consume B, B consumes C, but that means that indirectly, A actually has a positive impact on C. Or it's a double positive where species A benefits B, B benefits C, and then A also has an indirect positive impact. And so I'll give you a couple examples of these so that you're not just looking at A's and B's and arrows. So direct facilitation can be within species. And so an example of this is that a lot of grasses can share resources via their roots. And so these are photos from a salt marsh experiment where we were planting grasses in either a dispersed or an aggregated or clumped arrangement. And so, what happens when you plant these individual plugs of grass is that when they are clumped, they can share resources. Their roots are actually also putting oxygen into the soil. And so that's really helpful for salt marsh ecosystems that are constantly inundated. And so what that actually looks like a month later is that if you were to go back and look at the survivorship of some of these plots, the ones that actually have that were planted in clumps or aggregated do much better than the ones that were planted in a dispersed formation. And so here, we lost about, I guess, maybe almost 2/3 of the outplants. And this looks somewhat similar to what the previous slide looked like. A little bit less green, but still it's doing pretty well. Another example of direct facilitation are what I was talking about in terms of whole ecosystems. And so our coral reef systems are kind of an example of direct facilitation. So you have coral species. So in this example, we have a staghorn coral that is a foundation species, which means that it creates habitat. It's got all of this interstitial space that is able to provide refuge for a lot of different fishes. And so this beneficial relationship eventually leads to something that will look something like this, right? So these corals are actually creating this entire ecosystem. They're shifting the dynamics so that it's more amenable for other organisms to live within this structure that they create. And actually, in the process of living in this coral reefs tend to be pretty nutrient poor systems. The feces and poop that the fishes give off are actually very beneficial to the corals themselves as well. And so this then turns into a mutualistic relationship. An example of an indirect positive interaction. So this would be what is, some may be familiar with the term trophic cascade. So these are double negative interactions. And so one of the best examples of this is the impacts of top predators. So in this example, we have an otter that consumes and is a voracious predator of urchins. urchins are also a massive grazer on kelp. And so by keeping the urchin populations in check, these otters are able to facilitate the growth of kelp forests. We also have double positive facilitation cascades. And this is an example from mangroves. So mangroves are another foundation species that is creating habitat just by existing, essentially. Their prop roots that extend into the water are often colonized by what's known as a secondary foundation species. And in this example, we often see oysters that will colonize these mangrove prop roots. And so you can see this in this photo here as well. And all of this extra space that is created when these oysters colonize the prop roots then actually allows for all of these different smaller invertebrates, crabs, worms, snails, to be able to utilize these prop roots and create a ecosystem in and of itself. And so what's interesting about this, right, is that I've given all these examples of how these positive interactions are really crucial and form the entire basis of ecosystems. But we have this sort of odd paradigm in marine conservation. Oops, sorry, forgot I had that one. So these mangroves positively and indirectly facilitate this whole community of invertebrates. But as I was saying, we have this paradigm in marine conservation where we often are trying to minimize physical stressors, which includes reducing human impacts, and we're trying to suppress negative species interactions. And so that leads to approaches, at least in restoration, that look a little bit like this background photo, where you often see plantings that are highly dispersed. And this really doesn't allow for this utilization of those positive species interactions. And so this is kind of problematic because environmental restoration and ecological restoration is an inherently extremely stressful process. You're often trying to put back something that either was never there or was there in the past and for some reason is no longer there. And so the assumption may be that the area is no longer amenable to supporting whatever ecosystem used to be there in the past. And so this is a photo of what ecosystem restoration often looks like. Granted, I will also point out that this is in the context of a couple of experiments. So you can sort of see those same dispersed versus aggregated marsh plantings that I showed you in the past couple of slides. Oops. But also we have some oyster reefs that were built offshore of these natural marshes in an attempt to help buffer against some waves and to facilitate the growth of marshes behind this oyster reef. But if you look at this, this looks not very pretty. And so how do we turn this ecosystem into a lush, beautiful salt marsh? Well, one thing we can think about also is this relationship between positive interactions and stress. And so I'm going to walk you through this diagram. So on the x-axis down here, we have increasing environmental stress. And on the y-axis, we have the relative importance of different types of species interactions. Competition, so a negative interaction versus facilitation. And so what theory shows us and indicates to us is that as environmental stress increases, we typically see that the importance of a biotic interaction shifts from competition being important at low environmental stress to facilitation being important at high environmental stress. And then, of course, I will say that a certain amount of stress, there is no amount of interaction that can really continue to buffer against problems, right? Like if you're on the surface of the sun, there's no amount of facilitation that can prevent you from not dying. But that being said, what we want to think about is how these restoration sites are high stress and how we can utilize facilitation in these high stress scenarios. And so to just walk you through some research that I did when I was a PhD student in North Carolina, we were testing facilitation in seagrass restoration. And so there is a beneficial interaction in seagrasses that many people may not be familiar with because it's not one that's typically easily seen with your naked eye. So seagrass meadows are an incredibly important ecosystem for fishes, as well as a lot of crustacean and shellfish species, one of those being the hard clam. And so hard clams will settle in seagrasses and grow, and they're an important industry for North Carolina fisheries. What happens in this situation is that the seagrasses benefit the clams because they help their larvae to settle into the grasses. They provide some protection from predators for the clams as well. And the clams actually also will poop out pseudofeces, which are very rich in nutrients and is incredibly important for the growth of these grasses. And so you get this sort of mutualistic relationship between the grasses and the clams. And so what we did is we tested how we could incorporate clams into restoration of grasses. And we tested this with a different approach, several different approaches, where we either added clams with seagrass seeds to test interspecific facilitation, so whether clams can enhance seagrass growth from seed. And then we also tested this with both inter and intra-specific facilitation. And this is where we took adult shoots and looked at the relative effect of adding clams to adult shoot restoration, as well as the effect of planting configuration on restoration success. And so to sort of walk you through what this process looks like, we collected seeds from seagrasses in the early May. Of course, I'm losing track of the years now and dating myself. I guess this would have been 2015, I believe, May 2015. And you actually hold on to those seeds for quite a while before you can plant them because the seeds don't typically germinate until the winter time. So we go out in December or so and put the seeds out into essentially bare sediment in different mud flats. And we added them with and without clams. And then in the summertime, we also did some adult outplants and planted them in either dispersed or clumped configurations and with and without clams. And we just monitored to see how these plots did over time until Hurricane Florence, underlying theme of much of my work, it seems, led to our experiment end. And what was really cool about this project also is that we were able to partner with some local shellfish growers to get these tiny seed clams that were barely even maybe a centimeter wide to really look at how we could partner with aquaculture in order to enhance restoration or work towards conservation of these ecosystems. And so these next couple of slides are probably going to be the only ones that I'll actually, unfortunately, show you graphs. But I think they're pretty great because they're very stark and obvious graphs. So when we added clams to seeds, we saw that they massively increased growth. So in the presence of clams, the seagrass seeds were significantly longer in shoot length, right? So this is almost more than like a 100% increase in overall shoot length when we have clams. The amount of change in overall area from when these seeds started germinating to when the experiment ended was massive, right? It was a 400% increase in overall area when we had clams versus they didn't really increase in area all that much when it was just seagrasses. And then when we looked at below ground biomass, so these are the roots, it was also a massive increase, almost threefold in terms of the overall below ground biomass that was present when you added clams. Oops. We also saw just generally when we're looking at overall biomass, so these were the above ground would be, the green leafy shoots that the clams massively increased the amount of above ground biomass, the amount of leafy green shoots above the sediment surface. And they also increased the amount of reproductive effort. And so what you see here in these next two slides is the number of spates per shoot. So spates are sort of the reproductive organs. They're kind of like seagrass flowers. And in the presence of clams, you saw an almost more than twofold increase in the number of flowers, essentially. And the number of seeds that each flower was producing increased drastically in the presence of clams as well. Whoops, hello. There we go. And so the final sort of bit is trying to understand what underlies this relationship, what's driving these changes that we'd see. And so I mentioned in the beginning when I was talking about this relationship that the clams are pooping out pseudofeces, which are very rich in nitrogen, so essentially fertilizer. When we looked at the amount of nitrogen that was in these plots, both above and below ground, we found that in the presence of clams, the amount of nitrogen was more than 10 times greater than without clams. And then the below ground, we also saw a 5-fold increase in the amount of nitrogen when you added clams. And so I've shown you all those slides with figures of plots and graphs and whatnot, but seeing is believing, right? And so this is what just two comparative plots look like. And so on the left here, we have seagrasses without clams. And on the right here, we have a plot that was seagrasses planted with clams. And you can see pretty drastically the difference in how many shoots you see above ground in between comparing these two pictures. For the other half of the experiment, when we were restoring adult outplants, what was interesting here is that we actually saw no effect of clams. And so when you look at the clam comparison, you're looking at the dark blue versus the light blue. And then on our x-axis, we have an aggregated or dispersed configuration. And what I'll point out here is if you look across all of these plots, comparing dark blue and light blue, you don't really see a big difference in terms of the overall amount of shoots, the overall patch area at the experiment end, or the total change in these patches. But what you do see a big difference in is the configuration. And so I'll point out here, right, that this is proportional shoot change at the very end of the experiment between the very beginning. And you can see when it's aggregated, you have a positive change. So this means that these plots were growing, whereas when it was dispersed, they just consistently lost shoots. When we look at area, we see that the overall patch area was greater in these aggregated plots than the dispersed. And if we look at the change in area, again, we're seeing that these aggregated plots were increasing for the most part in overall area, whereas the dispersed plots were consistently decreasing in area. And so this is what those plots looked like. So you can sort of see that in these dispersed plantings, they look okay. They're not half bad, right? This middle one looks a little meager, I would say. But if you compare that with what it looks like when you were to plant them in one big clump, it's drastically different, right? Like it looks a lot more lush, and you can see that with your naked eye. And so sort of what we took from this is that a lot of these relationships are context dependent. And so you can't just sort of blanket say that like all positive interactions are great all the time. Instead, what was more the case was that interspecific facilitation, so that's relationships between the seagrasses and the clams, benefited restoration from seeds. And then intraspecific facilitation, so that's the planting configuration clumped versus dispersed, enhanced survivorship of adults. And so I'll just sort of finish up a couple with the next few slides, talking about how we can incorporate different beneficial partnerships in conservation outside of just seagrasses. And so you can do the same sort of approach with salt marsh grasses, right? I showed you those photos in the beginning where we planted marsh grasses in either dispersed versus clumped configurations. And this is an experiment done by Brian Silliman, who's a researcher at Duke. And he did this with collaborators in the Netherlands, where they planted them in this exact same different comparative approach and looked at growth over time. And you can see that the dispersed plots look like, I mean, there's grass there, it's not bad, but compared to the clumped plots, like this is a massive increase in the overall amount of above ground biomass that's in these clumped plots. In the Netherlands, you also sort of see that you lose a lot of plots, a lot of these individual outplants when they're dispersed compared to clumps. And again, this is another example where it's really important to understand context and which approach you should take and when. We know that physical stress tends to be greater at lower elevations in salt marshes. And so it'll be more important to utilize those positive intra-specific facilitations between those marsh grasses at mid to lower elevations. But actually at high elevation, physical stress doesn't tend to be super high. And in fact, that tends to be a situation again where competition may dominate the interactions there. And so it may be more beneficial to plant those grasses at high elevations in a dispersed configuration. And there are also examples from coral reef systems and understanding how all of these different interactions are forming this landscape or seascape. So if you look at these two diagrams, this is showing what happens when you include beneficial interactions in coral reefs. And so what we know, for example, is that mangroves can help to capture a lot of runoff from going out into seagrasses and coral reefs. Seagrasses also can help to reduce pathogens for nearby coral reefs, which is absolutely fascinating research as well. These fishes, when you have a really vibrant fish community, are going back and forth oftentimes between the mangroves and the coral reefs. They're again pooping out all of this nitrogen into an otherwise often nutrient poor system that is helping all of these different organisms to be productive. A lot of these fishes are grazers on algae, which can often compete with corals for space. And by conserving these grazing fishes, we can actually potentially increase the conservation of coral reefs. In a couple of other examples also from seagrasses, looking at those sort of indirect positive interactions that I talked about also. And so in this example, we have our lovely otter again, that consumes a mesopredator. So these are crabs, and these mesopredators also consume grazing herbivores. So these tinier little things like that are known as amphipods. And this sort of cascading interaction of indirect interactions from the presence of this top predator actually increases overall seagrass productivity. We see this in a lot of studies that have come out of California, where restoration of otter populations has really led to this shift in seagrass productivity in a lot of the embayments out there. But also there are examples of sharks. And so many people probably don't think too much about the process of sharks eating turtles, but they do in fact. And turtles are high grazers on seagrasses. There is in fact a seagrass that's called turtle grass. And if you are lucky enough one day to watch a turtle or green sea turtle grazing, it sort of acts like a lawnmower going through the seagrass ecosystem. And so by having these top predators, it keeps these grazing herbivores in check and maintains a high seagrass productivity and abundance. And then from a more humanistic perspective, we can also incorporate positive interactions in terms of the infrastructure that we use to protect our shorelines. And so some of this work comes from phenomenal researchers based in North Carolina, very good friends of mine, Rachel Gitman and Carter Smith, that have looked at different approaches to living shorelines. So these are approaches, shoreline stabilization approaches that are used typically to mimic nature in some way, shape, or form, but essentially they allow for organisms to colonize them and to use them as habitat, in comparison to things like bulkheads, where you really just have this gray infrastructure, vertical physical structure in the water column, and like you can't really use that if you're a fish, right? There's different approaches to living shorelines where you can either have only plants, or you can use plants like salt marshes and oyster reefs, or you can use things like granite rock to protect the shoreline. But what we've seen from a lot of this research is that this process of having these stabilization structures, whether it's an oyster or a marsh, actually helps to attenuate waves. It helps to prevent erosion for a lot of the ecosystems behind that living shoreline. These photos up here are from some of Rachel's work that has shown that living shorelines actually withstand the effects of hurricanes much better than bulkheads. And so this is an approach that we're seeing get a lot more traction across our state and across the nation in terms of protecting our shorelines. And so the last bit I'll talk about is some of the work that we're doing now in our lab that is incorporating secondary foundation species in restored ecosystems and living shorelines, actually. And so I want to give a big nod to our partners in this project, Bo Lusk and Britt Collins at the Nature Conservancy in Virginia, as well as Lexia Weaver and Todd Miller, who are our collaborators at the North Carolina Coastal Federation. None of this work could be done without these amazing NGOs that we partner with that quite literally do the heavy lifting of installing a lot of these restored ecosystems. And again, it's just a further example of the critical importance of partnerships. And so what I'll say on this front is that when most of us look out into a healthy estuary ecosystem, you may see something that looks a little bit like this, where you have salt marshes and oyster reefs growing abundantly throughout the estuary. And so what I'll point out in particular is that what you see in this picture is not just oyster reefs growing right next to salt marshes, but also oyster reefs growing on top and within, or sorry, salt marshes growing on top and within this oyster reef. And in this example, what we have is a primary foundation species. So these are common and abundant habitat-forming organisms that create locally stable conditions for other organisms. I've used this term to refer to some of those coral species. Seagrasses are also a foundation species. But in that example, what you also see is a secondary foundation species that would be the salt marsh grass that is also habitat forming, and it colonizes after that primary and further modifies that ecosystem's functions. It creates additional niche space. We can take this one step further. And in this example specifically, what we have is a complementary secondary foundation species. And so in this example, the grasses are a different morphology to the oysters, and they provide additional traits and services, right? So just even looking at this example, right, the grasses are tall, slender, oblong. They're photosynthesizers compared to oysters that are oblong filter feeders. They're reef builders. They tend to be much lower to the ground. But what you will also see are mussels will often grow with the oysters as well. And in this scenario, what you have are similar secondary foundation species. And so both of these species are filter feeders. They have similar traits. They're both reef building organisms, bivalves. And so, you know, they maybe potentially provide similar ecosystem services and functions. And this is important because based on whether or not this secondary foundation species is complementary versus similar, it's hypothesized that they'll support different types of ecosystems and different communities. So whether it is complementary typically means that it will increase the overall diversity of organisms that this ecosystem can support. And similar secondary foundation species are hypothesized to increase abundance, but not necessarily diversity of organisms. And then on top of just the community, secondary foundation species can likely increase resistance to stress. So there have been a lot of really cool studies that have shown that when mussels and marsh grasses grow together, they are more resistant to drought, for example. And I showed you those cute little diagrams, but these are photos of what this actually looks like, right? So there was the photo from the beginning of the graduate student pounding a pole in front of the marsh and oyster, but you do see these interactions occurring across many different systems, right? Mussels growing with marsh grasses, oysters growing with marsh grasses, oysters and mussels. This photo is multiple different species of seagrasses growing together. To the untrained eye, it may just look like a mix of green, but what you had here actually is one seagrass species. This branching one is known as rupia, and then this thicker one is zostra, and then there's another species in there that's so it's just, you see all these organisms interacting, and it's not always a competitive interaction. Oftentimes, these are beneficial interactions. But when we do restoration, it often looks a little bit more like this. And so, this is a pretty healthy looking restored reef off shore of this salt marsh. But what you don't see is any grasses necessarily planted within this reef, because oftentimes what restoration aims to do is to restore a primary species. And it's rare that you see multiple species co-restored, at least on top of one another. Oftentimes people will plant grasses behind each other, behind the reef, but not in the whole reef matrix itself. And so I'll give you a couple of just cursory results from some of this work that we've started this past year, where we looked just to see how common it was that secondary foundation species would naturally colonize these restored salt marshes, oyster reefs, and seagrass meadows. And so just some very cursory results. When we looked at both marshes and oysters, we saw that a lot of these restored sites actually had secondary foundation species. But what was interesting here is that in marshes, we tended to see a complementary secondary foundation species. So these were either oysters or mussels that would come in and colonize restored marshes. Whereas in oysters, or restored oysteries, we tended to see similar secondary foundation species. So that tended to be mussels that were coming in and colonizing restored oysteries. We didn't see a ton of secondary foundation species in restored seagrass meadows, but what I will say is that most of those surveys, all of those surveys occurred in Virginia, which does tend to be a bit of a monospecific area. And so that is a ecosystem where there only tends to be one seagrass species that dominates. Likely, we would expect that there would be more secondary foundation species in North Carolina, where we do see that mix of different seagrass species, as well as macroalgae that grows on the grasses. And if we just do a quick comparison to look at productivity and how that changes with the amount of secondary foundation species, we actually see that as you increase the amount of secondary foundation species within an area, it did look like it was positively correlated with the amount of live oyster cover. And so this is, kind of a good indication that possibly, even if you don't co-restore, you will see these beneficial interactions take shape over time. And so a couple of photos from some of the sites that we looked at, we saw mussels coming in to restored marshes. This is a photo actually from my dissertation work where you did see macroalgae that was colonizing these grasses towards the end of the season, and potentially increasing the amount of habitat and niche space for different organisms. You actually managed to see, amazingly enough, grasses growing through these oyster bags that are filled with shell and are, I guess they're probably around like 5 to 10 pounds in weight. So it's amazing to me that they can grow through all of that oyster shell and through that bag. And then in this photo, you can see a lot of mussels that have colonized this oyster reef as well. And so these secondary foundation species are coming in on their own, but sort of the next couple of steps that we're moving with this research is to look at how you can utilize this from the ground up. And what happens when you do try and co-restore and you plant maybe grasses in between these oyster bags, or there are different approaches to oyster restoration that actually have more open space, that you can plant grasses in those open spaces, or if you planted marsh grasses with mussels, and take the different approaches to co-restoration when you begin it at the very start. And with that, I'll just take any questions.
Fascinating stuff. Everybody, let's give Dr. Zhang a great big round of applause wherever you happen to be. If you hear clapping down the hall from your office, let us know. Incredible stuff. Love this research. Absolutely like crucial, critical stuff for the work of restoration, I feel like. And just also really neat to see all of the different ways that these species interact with one another, even in the smallest of test plots. Mm-hmm. Like in 1 meter squared, that you could have so many interactions happening between even what seems like a small number of species. Mm-hmm. is pretty cool stuff.
Yeah, I really think it's fascinating because, right, like these interactions can be at very, very small scales, right? Like between a single clam and a seagrass seed. But then they can also be these massive habitat forming interactions, like between the oyster reefs and salt marshes, right? Like they occur at multiple different spatial scales, which I actually think is really beneficial too, in terms of like, if you are a practitioner and you maybe want to incorporate this, you don't necessarily have to do it at a massive scale. You could choose a smaller spatial scale or a smaller species interaction in terms of space. Yeah.
Especially when, I mean, you mentioned some of the NGOs that you've worked with and the nonprofit partners who do a lot of the restoration work. Like, there's not always a huge amount of money to do massive We can't restore the entire coastline of North Carolina from all of the development that's happened, for example, or the erosion that's happened. But if we can take advantage of good research like yours and really build in the kind of communities that are going to give us the best benefits, right, then what resources we have to do this conservation work goes even further.
Yeah. And there's different approaches, right? In terms of planting the grasses in a clumped like configuration doesn't cost anything. That's just a matter of changing the way that you go about doing things versus like having to buy a ton of extra materials. The group that I do the work in North Carolina with, the Coastal Federation, does a lot of really great marsh restoration work and living shoreline restoration. And they'll tell this story. I'm going to tell this story from Rachel Bassesi, who is one of their community outreach coordinators. And she talks about how when we go out, a lot of times they'll work with different groups that will volunteer their time. And she'll say, like, plant these grasses closest together. And she tells this story about how they were working with a elementary school group, And towards the end of the day, the kids were they were done. They were not having it. They did not want to plant any more marsh grasses. And so they took this whole tray of grasses and just planted it all together instead of like planting them in like a more dispersed way. And that big clump of grasses that they planted did really well. And so, yeah, I think it's just really amazing to see how just small changes can be used. And even like, a kid being tired and just wanting to plant all of the grasses together can actually have a really beneficial interaction.
I love that story. That's great. Okay. There are some questions for you in the chat. You ready to take some audience thoughts?
All right.
Okay. So the first one, Aaron wanted to know if the clams that you used in your study plots were lucinids or venerids.
So they were not lucinids or venerids. And so actually, when we did it they're both the same species. So they're Mercenaria, which is known as a quahog, but they were different sizes. So lucinids are a type of clam that has been shown in a lot of different studies to actually have this symbiotic sulfur reducing bacteria that's beneficial for seagrasses. We don't see them super, super often in North Carolina, but we do see a lot of hard clams. And so, it's a slightly different interaction there. And so it's probably not a sulfur reducing mechanism in our example, but yeah, that's another really great symbiotic relationship that we see with different grasses.
Excellent stuff. The next one, Amanda was asking, the clams had huge benefits to seagrass from seed. Did you also look at whether there were quantifiable benefits to the clams in the different scenarios?
Yeah, so we wanted to, but actually, and I wonder how much of this is linked. The clams didn't always survive. And so we often did see in each of those plots around like 2 to 3 clams dying. And I do wonder actually if like the process of those clams dying was also releasing nitrogen and phosphorus into the sediment. So we weren't able to specifically look at that. But yeah, that's another avenue of research that's really important. I think there's actually folks in Virginia who are looking at that to see how seagrasses benefit the growth of clams and whether or not they protect against predation.
Okay, fascinating. I was kind of curious. You had mentioned early in the presentation that these restoration projects can be stressful on the things that you are using for the restoration. Right. And so thinking about the grasses and the clams and the stress that they're going to be under, even though you're trying to reduce the overall amount of stress in this environment or in this ecosystem by doing restoration, I thought was an interesting, a paradox kind of a thing.
Yeah.
But okay, more for you. Let's see. Would it be correct to say that the best long-term plan would be planting aggregated seed grasses from seed alongside clams? Would that be beneficial for both the youth and adult grasses?
Well, that's a great question. It's such a hard question to answer because so oftentimes ecosystems are really context dependent. So for our system, maybe that is the best approach where planting from seeds, adding clams at that beginning kind of gives that little bit of oomph at the very beginning. And so that may be one of the approaches that we should take in North Carolina. In situations that may not be as environmentally stressful, it may not be the case. I guess I'm reticent to say that there is a one size fits all situation. Because I know there are situations where clams and seagrasses could actually be competing for space, so if it's in a situation where you're planting really dense grasses, then maybe adding those clams is not as beneficial. But I would say if I were able to go out and do this on a massive scale outside of just those tiny plots, I would definitely test that.
Excellent, thank you. And Jessica wants to know, when restoring a habitat, will secondary foundation species naturally populate again once the primary foundation species are established? Or I guess, do they need to be planted alongside the PFS?
So sort of what our initial surveys were showing us is that they will naturally colonize. Some of those restored sites vary in age from anywhere between 2 years ago to 10 years ago. And so that's another aspect we're interested in looking into, is how the age, how that changes with age, but also proximity to other natural sites where you do see secondary foundation species. And yeah, some of the experiments we want to do in the future is going to look into what happens when you do it and you actually have to put in those secondary foundation species at the very beginning, just to see if it gives that extra oomph, right? So we do see this positive relationship when they naturally colonize. So can we just go ahead and like speed time along and instantly get to that like higher density of primary and secondary? And does that mean that maybe the outplants will do better overall?
Excellent stuff. Thank you very much for taking some questions.
Yeah, of course.
Let's see here. You know, I'm kind of curious about let me think about this for a second, because North Carolina's coasts, I feel like we have a lot of development. We have a lot of problems with sea level rise incoming and coastal erosion problems. We see beach houses collapsing into the oceans all the time. And it seems like we need pretty massive restoration efforts in order to get our coastlines back to some kind of, I don't want to say a totally natural state, because I don't believe we'll get there. We exist in these landscapes too. Maybe that's it. How do you see people continuing to exist in these ecosystems when we still like bulkheads and we still like jetties, and but the research continues to show that we really need to be working on these living shorelines?
Yeah, that is a great question. I think there's a lot of that's tied in with that. Understanding coastal resilience, not just from an ecological perspective, but also from a human and community perspective, is really important. And I think a lot of it is education. Some of the great work that the museum is doing, having this seminar or this lunchtime discovery series, and educating folks on these interactions and living shorelines, that this is an approach that you can actually use. A lot of people, homeowners, maybe weren't aware of the fact that this was an approach they could use to stabilize their shorelines. And I believe some of the really cool work that Carter Smith has done has shown that one of the best indicators of whether or not someone installs a bulkhead or a living shoreline is if your neighbor installs it. And so a lot of this is just continued communication of different approaches that we can use, in terms of understanding how we build a community to be resilient to climate change. We do see indications that oyster reefs can keep pace with sea level rise. So they'll continue accreting sediment. The work that the researchers have shown where marshes and these living shorelines can help to buffer against hurricanes. But yeah, there are some massive approaches that we need to consider. But all of that needs to be done with community investment and buy-in, right? And so, yeah, there's a lot that needs to be done.
Absolutely. We've got 1 minute left in the show. I did want to ask you, did you go check on your research plots after Hurricane Florence? And is that what ended your research, or was that just sort of like we can be done with it at this point? Did you go look and they were just gone and you were like, well, that's the game?
I did look afterwards and they were gone. And I was like, and that's another aspect we need to think about, right? Is how do we make these these restored projects resistant to storms. I'm sure it didn't help that my plots were much smaller in size, but actually, that makes me think about there's some research being done in Florida where they're planting dune grasses and doing similar things where they're dispersed versus aggregated. And this was at a site that was hit by a hurricane last year, and those sites where they were planting in aggregated configurations were able to actually come back after the hurricane, but when it was dispersed, they did not. And so, yeah, I think there's different approaches, and doing things perhaps on a bigger spatial scale helps.
Fascinating stuff. Well, Stacy, thank you for being on the program today and sharing your insights and expertise with us.
Thank you so much for having me.
Really appreciate it. We'll probably have you back again real soon.
All right.
It sounds like you're doing some great stuff.
It's all because of the people that I work with. So.
And viewers, thanks for tuning in to today's edition of the Lunchtime Discovery Series. I know that you enjoyed it. We had a great time. I know you learned something new. And so I hope that you'll come back and join us again here at the museum's YouTube channel. We'll be back here again next Wednesday at noon with another presentation. And from the looks of things, we're going to stay out on the coast for a presentation about flooding with Dr. Catherine Annard. So don't miss out. We're going to be right here again next Wednesday at noon with another great program. Make sure that you hit up naturalsciences.org. There you can find lots of programs and events just like this one. And if you want to keep up with what's happening with the Lunchtime Discovery Series itself, make sure that you head over to eenorthcarolina.org. You can also see an archive of past presentations there. So there's a lot of interesting stuff that you can learn and take in over at their website. Many thanks to Stacy Zhang, Dr. Zhang, our guest for today, to the folks over at the Office of Environmental Education and the museum's digital media team for helping put all of this together and getting this broadcast out. We will see you all again next week. Bye, everybody.